Hello everyone, my name is David Tabb and I'm an American bioinformatics researcher. I specialize in proteome informatics, but I thought it would be useful to do a little bit of an introduction of myself for this Bioinformatics Exchange Day. I grew up in Missouri, which is in the middle of the United States, but I went to college down south in Arkansas, which isn't that far away, but culturally in the United States it can feel pretty far away. I decided to major in biology. I'd taken a lot of biology when I was in high school and wanted to continue that as I moved into toward a more professional direction, thinking about biology, sort of bench biology as my career. So I decided to major in biology, which was a new department for Arkansas at that time. They had a pre-existing microbiology department, an environmental science, and a pre-med track, and those were all boiled together into one biology program. So in 1996, I finished my degree there, and I decided at that point that it was really important to me to have computers be part of my research. So instead of just going to a straightforward biology or biochemistry graduate program, I decided to go to a molecular biotechnology program that was being started for the first time out at the University of Washington in Seattle. Lee Hood was creating a department there. So I moved to Seattle in 1996 and started my graduate work there. And that was really where I first came into contact with mass spectrometry. I decided to join the lab of John Yates, who, although I didn't know it at the time, was a huge pioneer in this field of proteomics. And as a result, I got just kind of the best education I possibly could have in how to deal with the data from tandem mass spectrometry in the field of protein identification. Now, our lab moved about halfway through my graduate training. So I finished my degree while I was at Scripps Research, but my PhD is actually from the University of Washington. I graduated from there in 2003. Now I decided that I was really interested in what was going on in the National Lab Program. This is not really an academic environment, but more of a government laboratory environment for the United States. So I moved to Oak Ridge, Tennessee. That's in the eastern part of my state. And I worked as part of the Genomes to Life program at Oak Ridge National Laboratory, where I was part of the Genome Analysis and Systems Modeling Group. Mostly my work was in connection with the mass spectrometrists who were working over in life sciences, in the chemical sciences division rather. So I worked with them for a couple of years, and then I got the opportunity to come to Vanderbilt University as an assistant professor. And that was a really hard opportunity to turn down, so I came to Nashville for that in 2005. So for the last 10 years, I've been here in Nashville working at Vanderbilt University. It's been a really wonderful experience. I made tenure in 2011 and I was really excited about that. But more recently, the research climate in the United States has become kind of unpleasant, with a smaller and smaller pool of dollars being competed for by larger and larger groups of people. So I thought it was time to make a change, and I'm really excited about the opportunities at Stellenbosch University. Being able to work in an area like tuberculosis that affects the lives of lots and lots of people and takes a lot of lives for people who, did nothing worse than growing up in a poor area is, I think, a really strong motivation drive for me to move my research to that area. Now, I've been working in colon cancer for the last 10 years or so, but the technologies have really been my emphasis, and those technologies are just as applicable in tuberculosis as they have been in colon cancer. Once I arrive at Stellenbosch, my plan is for my research program to take part in 3 different areas. The first is in support of the Medical Research Council SHIP program. As I understand it, it's more or less a translational science infrastructure program. Now, there are lots of researchers within the MRC SHIP program who are doing tuberculosis research, and many of them are generating fairly substantial data sets, but many of them aren't so sure what to do with them. Now, I think that support is going to involve a lot of pathways outside of the area of proteomics, and so I'll need to brush off some of my skills for dealing with microarray data sets or working with next-generation sequencing data sets. But the upshot is that somebody with a data set that they've generated under a SHIP TB grant can phone me up and say, I've got these data, what do I do with them? So, since they're paying the bills, I'm going to be giving a fair amount of effort to making sure that that all happens. On the second front, I'm very interested in supporting the expansion of the role that mass spectrometry plays, specifically biological mass spectrometry plays, within Stellenbosch University, within Cape Town, and within South Africa as a whole. Within Stellenbosch, I think the mandate is pretty clear. You have your Orbitrap fusion instrument, you have some great people running those instruments. It's making sure that people understand what they can expect when they go into work with the mass spectrometry data. What kind of result will they get? Being able to interpret those results in the context of their research is something that I care very strongly about. So I see myself working very closely with Murray Flock about getting those data into the hands of people who need them. So the second area as we move out to Cape Town is to be able to work with people at the Center for Proteomics and Genomics and University of the Western Cape and so on, to be able to work with folks in that area who have mass spectrometers and need to be able to interpret their data better. That's really the area that my research niche has farmed out the best. But as we look even more broadly across South Africa, it's certainly clear that when proteomics labs are working cheek by jowl with other proteomics laboratories, their skills get a lot better. And I'm really interested in seeing if South Africa can develop a program, something like the one that recently was such a success in Spain, called ProteoRed, in which different laboratories were given common samples and they all worked out their best analysis of them, and they put the results together and got to see how each lab compared to the others. If some of the labs are lagging behind, people from the labs that are functioning a little better can help them to figure out the ways to optimize their processes to get better data from their experiments. I think that's really in the interests of everybody. So I believe biological mass spectrometry has a very substantial role to play in South Africa, and I'm very intent on making that come to pass. The third area that I want to work in is bioinformatics education. Now, I realize that in the British system there are not a lot of courses per se that students take when they're in graduate school, but I know that there are students taking their honors year over at the Stellenbosch main campus that can usefully gain from an exposure to bioinformatics at that point. And there are students who come into programs like the University of the Western Capes each summer from across all of Africa that could use better training in the area of mass spectrometry bioinformatics. So I'm really looking forward to taking part in that kind of basically graduate level training. But the other piece that I'm interested in is finding a better way for biotechnology information being created at places like Stellenbosch can percolate out to the very much broader community of South Africa. And that may mean that I need to learn a little bit of Xhosa or Zulu, but probably it means I need to find a collaborator who can speak those languages to put together things like YouTube videos or even cell phone message systems where people can learn a little bit about what biotechnology has to offer them, because we certainly are doing work that's relevant to them. So I'm really excited about those opportunities. I think it's a very natural question, when will I arrive? I thought that this would all move a little faster than it has actually, but we're trying to do the best we can to make this process move along rapidly. I've been borrowing from the experience of Gerard Trump, who recently went through this experience himself. So I see this process as 3 steps. The first is SAQA, the SAQA, the South African Qualifications Authority, needs to judge that my diplomas are real. We've completed that step. SAQA has agreed that my bachelor's and PhD are real, so that step is completed. The second one is membership in SACNASP. Being part of the Association of Natural Scientific Professions will mean that I have a letter from a South African government institution that attests to my critical skills and gets me through the visa process. So I have applied for membership in SACNASP on July 13th, and on July 22nd, all the referee reports were completed. Hopefully, sometime, anytime, I will hear back from SACNASP that my membership is complete. Once that's in place, we can finally trigger the third step, which is my department to the Department of Home Affairs in the government. I send them my BI 1738 form along with a whole bunch of other information that I've already collected here stateside that will be passed to the South African government. At that point, I need to travel to the nearest consulate, either New York or Washington, and do an in-person interview, I believe. And then, hopefully, the temporary residence permit will be granted and I can fly to South Africa at last. But we're really waiting on those last 2 approvals. SACNASP says that they can take anywhere from 3 months on to evaluate a membership application. It's already been a month, so I've been poking them a little bit to see whether that can be wrapped up a little more quickly. The other piece is that the house I'm standing in needs to be sold. And I've been on the market now, and I've had a pretty good chance to sell it, but the contract fell through. But I had a lot more views this week, and I'm hopeful that another contract will be in place soon. So, if you were to ask me, when will I be in South Africa? I think the soonest that I could reasonably hope to be there is October. Hopefully I'll find out more from SACNASP soon that will let me predict that date a little more closely. But for the meanwhile, I'm trying to get myself prepared by learning as much as I can about my new country, and I'm looking forward to being there with you. So finally, I would like to talk about some of the services that I hope that my laboratory will be able to offer. The first one that I think everyone thinks of in connection with who I am is protein identification services at very large scale. I've been part of a lot of experiments that have included months of mass spectrometry time, all included in a single experiment. So being able to do protein identification at that scale is something that my lab is really specialized in. We have the ability to use conventional database search techniques, some algorithms of which we've actually created ourselves. We have the ability to do sequence tagging identification. This is very good when you are looking for post-translational modifications of proteins. We also have the ability to do spectral library identification, which is very valuable when you spend all of your time focusing on one or two particular organisms. So if you have a library of spectra that you've identified from them in the past, you can leverage those in order to identify new spectra that you've collected. So we've created the ID Picker protein assembly that is very powerful for pulling together the results from lots and lots of experiments all in one assembly to help make coherent very large reams of data. Also to connect them externally, in the case of proteogenomic experiments, to genomic information that we have available. So the next piece that I would talk about is that we have an emphasis in quality control for proteomics as well. My laboratory has been working on statistical models by which we can recognize outlier experiments. It might be that you think that an experiment produced far fewer spectra for a particular protein, and you might judge that that one was downregulated. But your conclusion about that protein would differ if you realized that all proteins had lower spectral counts in that data set. So being able to recognize these outliers so that people don't confuse technical variability for biological variability is something that I care about very strongly. I've also developed an interest over the years in metabolomics, being able to interpret, for example, the tandem mass spectra from phospholipids. So being able to extend the use of mass spectrometry beyond proteins is something that I care about as well. Now, as I've mentioned before, I need to be cognizant that not everyone's working with mass spectrometry data. I would fully expect that I'm going to be dealing with some microarrays and certainly a lot of next-generation sequencing data. For that, I intend to get myself familiarized a lot more with the Galaxy framework for creating whole pipelines of analyses in next-generation sequencing. And also working to an increasing extent with Bioconductor, which is a tool that I've used a little bit from the XCMS library for metabolomics, but being able to apply that in the context of microarray and other data sets. So I'm really hopeful about about broadening out from my emphasis just in protein identification and post-translational modification identification to a much broader cut across what's available in bioinformatics. The other thing that I'm excited about is that in my negotiations with Vanderbilt University, I was able to take the computers that I had been using in my laboratory for use in South Africa. So I'm hoping to show you a photograph here of a set of computers currently waiting in my attic, ready for shipment to South Africa along with the rest of my stuff. So this is including a cluster of computers that I have used for compute-heavy processes. So there are 8 computers, one that has a very large amount of RAM that I can use for processing jobs in Linux that would otherwise take a very long time on an individual computer. I also have a file server that I'm bringing that has 8 2-terabyte drives in it, so kind of a workgroup file server. And of course, a few desktop workstations as well for me to use and people who are working with my team. So I'm really looking forward to being there, and I hope that this little video has given you a little chance to get to know what I do and what I'm hoping to do there in Stellenbosch. Thank you.